So the next thing we're going to start looking at is this is where we're going to get into the more of the biology part of the course and the biochemistry. You're going to note that I'm going to have a pretty different perspective toward as opposed to like your biology. Okay? But I'm going to try to keep it somewhat on the same level as that. So you're not going, what in the world is this, Dr. Clausen? So you have some sort of relation to it. Okay? Okay, so the things we're going to look at first is your proteins. Your proteins are many of the workers, quote unquote, of your cell. They're a lot of the function that you think of as the functions you do and you exist by are actually protein or protein complexes. Okay? Some of the structural is also proteins as well. Though there are, of course, carbohydrate components to those proteins, and of course, lipid components to the, not proteins, but structural components. So carbs and lipids actually take a pretty significant chunk of that structural component in your body. But the workers, the actual work getting done, is typically protein complexes, with a little bit of RNA in there as some of the complexes as well. All right, so let's talk about the amino acids. These are going to be the building blocks of your proteins. Poly means many. Meros means part. And so proteins are an example of a polymer, and that's how we combine that in English. We actually modify it slightly from the old Latin. Okay? And so has anybody ever heard of a polymer before? Yeah, like plastic, right? Or maybe your clothes. Your clothes are polymers. Okay? In the case of proteins, the monomers, which are the single units of polymers, many unit chains, are actually going to be your amino acids, and you've probably heard of those amino acids before. Okay, you can actually refer to these as polyamides because that's the linkage. Okay? There's 20 different commonly occurring amino acids. Proteins can contain as many as 50. As few as 50, and as many as thousands of amino acids. These things can get extremely large and extremely long, actually. They're typically not actually folded out into chains. They're typically folded up into very specific structures. And the structure of a protein is a large portion of its function. Okay? Kind of like you can always tell when you're looking at a copier, you can always tell when you're looking at like a crane, because it has to have a certain structure to do what it does, doesn't it? Like you can always tell when you're looking at something that grasps something, because it has to have a structure that's conducive to that. Okay? The ordering of the amino acids in a protein is going to determine both the structure and the biological function, though it is not only the amino acids that does this. You have other things that are going to be included. For example, you need metals in many of the proteins for them to fold correctly, okay? And so like magnesium is actually quite important in this process. A lot more important than you may have realized, all right? Okay, so proteins can transport oxygen in blood. That's probably the one you know the best, which is hemoglobin. Everybody's heard of hemoglobin before? Yeah? Okay. Anybody heard of myoglobin? Yeah? Yeah, maybe? Okay, fantastic. What comes out of meat, by the way? When you get like a steak and it's rare and the quote-unquote blood is dripping out, what is that? Is that actually blood? No, it's actually myoglobin. Yeah, it's not actually hemoglobin. So, it's not blood. Proteins, again, can do things like serve as the main components of skin and muscle. Your muscle especially is a protein complex. They can defend our bodies against infection. Our immune system is mostly composed of protein, though the identifiers on our cell are carbohydrates, like we discussed, right? They can direct cellular chemistry. They do this through being enzymes. For a long time, it was actually thought that all enzymes were protein, period. And there was nothing else. There has been found some enzymes that are RNA, and mostly RNA. And we, in fact, have ribosomes in our bodies that are large part RNA, but they do have protein components. Okay. They can control metabolism as hormones, though there's other hormones that are not protein complexes. However, hormones all have to bind to protein complexes to do their function in our body. Okay. The amino is actually indicate the protonated amine. So when we look at amino acids, that actually indicates the NH3 plus, okay, which is the amine NH2, right, neutral, with a proton on it. Notice that NH2 neutral was a base. It's a weak base, okay? The other part of the amino acid is the acid. And what that indicates is that has a carboxylate. Now, carboxylate is actually the conjugated base of carboxylic acid. And so when you have an amino acid, it actually has a base portion and an acid portion that actually loses the proton and protonates the base portion. Okay? This is typically referred to as a zwitterion, meaning two-side ion. Overall charge is 0, but it has two sides where the charges are different. Now, some amino acids are not actually zwitterions at physiological pH, and that's because these things have side chains. Okay? Some of the side chains are actually basic or acidic. And so, but when we're looking at the main portion of the amino acid, that always loses a proton, gains a proton at physiological pH in your body. Okay? All right, so amino acids, these groups are going to be bonded to a central carbon. This carbon is referred to as the alpha carbon. You already recognize that terminology because that was always the carbon that was next to the functional group. And so this carbon in the middle is always next to two functional groups, these ones. And so it's referred to as the alpha carbon. This is also the carbon that has the side chain connected to it. If we look at our amino acid, this is the overall drawing of an amino acid. This is the side chain that creates those 20 different amino acids. Okay? All the 20 different ones just have different groups on this side chain. Some of them do actually wrap back around and bond to this and can create intramolecular bonds. But that's the difference between all the amino acids. Those 20 amino acids, they are the side chain right here. So see that R group? Everybody's comfortable with the R group now? Yeah, see? That organic chemistry portion actually had meaning, didn't it? Yeah, we can relate and talk about biological compounds now and actually understand what the heck we're looking at. Okay? This one's the alpha carbon because it's always next to all the functional groups, isn't it? Yeah. Notice right here we have the protonated amine, that's the amino. And over here, the carboxylate acid, that's the acid, right? It has lost its proton in that case because we have a base right next to it. Okay. What do we know about this alpha carbon? Is there something special about that alpha carbon right there? Got it. That's a chiral center, isn't it? Has 4 different things around it, right? And so you can have different chiralities of amino acids. Those chiralities would be R and S. And so the alpha carbon is going to be bonded to that hydrogen atom right there. And it's going to be designated by an R for the side chain right here. And anytime you look at amino acids, you can always have the RS conformations or DNL amino acids. Okay? And interestingly enough, all life only uses one conformation. Has anybody ever grabbed like an amino acid powder? Like alanine powder or anything like that, tyrosine? Pretty common for if you're trying to like bulk up or you're trying to do specific exercises. Very useful for runners, actually. So those are actually the conformations. You can actually get these powders in conformation pure with only one chirality. You can also get them where they are racemic, meaning they have both chiralities. If you get racemic powders, 50% of the powder is just going to pass through you and you cannot use it. Okay? And so people oftentimes will pay 75% the cost to get a racemic mixture. Was that a good purchase? They're usually about 20% cheaper. Was that a good purchase? No, you paid 80% the price to get 50% the product, right? That was a terrible purchase. Really common if you go into the health food stores and stuff. You'll see the slightly more expensive one, same amount, slightly cheaper one, same bottle size, and you got a cheaper one. And then if you look carefully, it'll actually say racemic on it. It's a trick that gets played. So right here, these are the chiral centers. This right here is the L amino acid right there. This one's the D amino acid right here. Notice we're in the same Fisher projections that we were looking at before. So these are coming out towards you like this, going away from you above and below. Okay. And so the L amino acids are actually the building blocks of proteins right here. Those are the ones that you use in your body and you can actually function with. The D amino acids can occur in nature actually, but rarely occur in proteins and are essentially useless to you. Okay? There are like bacteria that can actually digest these and convert them into the form they want. And yeah, but you, not so much. So the side chain, or R group, this is going to give you each amino acid, its unique identity and characteristics. These are what those amino acids are named after. Remember, all the amino acids that you're going to typically use and see and are going to be the essential amino acids for a human are going to be the L amino acids. Okay? There are 20 amino acids that are found in most proteins. Does that mean there's the only 20 amino acids in nature at all? No. You can actually have more amino acids, but these are the ones that would occur in proteins in a human. Okay? And in most animals, to be honest. There's very few organisms that would have more amino acids than 20. Okay? At least that you probably care about. There's also a whole host of synthetic ones, and now there's even a thing where they're thinking about making mirror organisms, where all the chiralities of everything would be inverted. So everything would be the opposite chirality. People are not up for. Yeah, because they could be like toxic to all living beings, or they could interact not at all. We don't know. There's 9 families of organic compounds that are represented in these. There's the alkanes. Those are the hydrocarbons. You probably saw that carbon skeleton. There's the aromatics. There's the sulfides. There's the alcohols. There's the phenols, the thiols, the amides, the carboxylates, and the protonated amines. We've already seen the carboxylates, protonated amines, and the hydrocarbon portions. The other ones are going to be on those side chains. Okay. The properties of functional groups are going to allow them to be classified. They can be classified as polar and nonpolar, which is the primary classification that's going to matter the most. That's actually going to create the parts of the different protein that cause it to fold, actually, and create the structures, okay, for the most part. It's the biggest driving force. The polar amino acids are going to be further classified as neutral, acidic, or basic. So if they're neutral at physiological pH, they're going to be the neutral ones. If in the body they tend to be acidic, they're going to be acidic. And then finally, if they're basic in the body, they're going to be basic. Okay, so right here we see a hydrocarbon one. This one has a hydrogen. By the way, does this one right here have chirality? It doesn't, though. Yeah. So this one right here does not actually have. Okay. This is the only one that doesn't have chirality, by the way. And so that's glycine. This is the 3-letter abbreviation, and then next that is the 1-letter abbreviations. Okay? So typically, if you were going to represent a protein, you would do it by 1-letter abbreviations, because they're very long. Okay? If you're representing parts of proteins, little tiny fragments that you want to talk about, you could use the 3-letter representations. And finally, if you're just talking about the single amino acids, or maybe 2 or 3 of them, you can use the full names. Okay? But imagine trying to write out full names for 10,000 of these. Anyone want to do it? No. Everybody, my eyes went on is like, don't assign that. You imagine. This one's alanine. Everybody probably recognizes alanine, right? You guys have all heard of alanine? No? Okay, and so this is the hydrocarbon right there, has CH3. This one is valine. Notice the V here. Yeah, that's that isopropyl group, isn't it? Yep. This one right here has the isobutyl group, by the way. And that's leucine. Isoleucine right here. Notice it's a rearrangement of the leucine. Cool, huh? Ooh. It's like the organic naming I taught you was meaningful. And that's how they actually base these names. This right here actually has an aromatic group right there. And so this is phenylalanine. Note, by the way, it's like alanine, but it has a phenyl group right here, which is the aromatic substituent, the benzene substituent. Okay, these are again the nonpolar ones because if you look at this side chain, it's nonpolar. This is proline. You can see it makes a little P here. This one's methionine. That's the methyl group off of a thiol group, right? So methyl, methionine. This one right here is tryptophan because it looks trippy over here. This is the polar amino acids. These ones are neutral. And so you'll see like they have alcohols, phenol, thiols. These things are neutral, right? This is actually a secondary alcohol. It's actually kind of easy to remember because it has threonine, 3 different groups on this. And this one is serine because if you drink alcohol, you're serene, right? I'm not proposing alcohol. Okay, and so this one's phenyl group right here. This is tyrosine. This one's cysteine. You guys may recognize cysteine as being heavily involved in like thiol bridges or things like hair. Okay, this is actually what causes curly hair, in case you're wondering. This is your amino acids that are neutral again. The ending continuation of it, because I can't fit them all on one page. And so asparagine, glutamine. Notice the enes because they have the amides here with the nitrogens. This right here is going to be your polar amino acids. They're acidic. And what you'll notice here is that these are the conjugate bases of your carboxylic acids, aren't they? Okay, so these have carboxylic acids on your side chains. And so that's aspartate. Note that, again, this is carboxylate, so the ending is 8 to tell you that it's a carboxylate, isn't it? Okay. And so we have the carboxylate right here with only one CH2 between, aspartate, and this one's going to be glutamate. Interesting side note is aspartate is actually modified to make one of the artificial sugars. Okay. This one's the basic ones. And so this is histidine. You guys may have heard of histidine before. And so that's going to be basic. Notice it's the conjugate acid of a base. So when you threw it in, that physiological pH solution was a base. It reacted as a base. Now you got your conjugate acid left. And over here, this is lysine. And then this is arginine right here. Notice this one actually has 2 nitrogens at the end there. It's kind of an interesting side chain. Okay, so the 10 amino acids that have been designated with an asterisk are going to be called essential amino acids. This is actually because they cannot be synthesized in the human body. They actually have to be made, obtained in your diet. If you don't get them in your diet, you just don't get them. You don't have them to make the enzymes, the proteins, and they're going to cause you issues because you won't be able to make those enzyme proteins. Okay? Possibly leading up to your death. So, 2 of these amino acids, arginine and histidine, are actually essential in the diets of children, but are not actually so essential in adults. We can actually go for fairly long periods without them and have very low levels of them. Okay? They're just not as needed once you're grown up. Proteins that contain all the essential amino acids are going to be known as complete proteins. Most of these are going to be animal proteins because animals have the same amino acids in their diets they need as us. And so if you eat all the parts of an animal, you're going to get pretty much everything, right? Now, that does not mean that's an efficient way of getting them. Some of the more efficient ways of getting them are going to be soybeans. That's why it's a very popular source of protein, is because it's actually complete. That's not to say that it actually gives you the correct ratio of protein, of amino acids, but you can get them all from it. Okay. Eggs, milk, fish, poultry, and meats of basically all variety, as long as it's an animal meat, is going to give you a complete source of proteins. Again, complete does not mean in the ratio you actually need. Okay. Okay. So you can end up, if you eat one of these things over and over and over and never eat anything else, you can end up with a lot extra of one of amino acid than you need and a lot less than you need of another. Okay? And you might have to eat a lot more calories than you necessarily need to get everything. Okay? That's why it's better to have like more of a mixed diet, right? And obviously you don't actually need milk. You can do other things, right? Okay. By the way, a little interesting side note here. So does anybody know anything about like healthy foods and health, what you're supposed to eat? Anybody ever heard of the food pyramid? Okay. Okay. And so the food pyramid is not taught anymore, right? I hope. Was anybody taught the food pyramid? Kinda? Really? Wow. Kind of. Kind of. So during the Obama era, they changed over the food wheel. And the food wheel is so much better than the food pyramid, but it's still pretty off. You can interpret it correctly, but most people do not. Let's put it that way. Okay. The food pyramid is just totally wrong. Like absolutely, almost the reverse of what you should eat. Yeah. Like the amount of carbohydrates you need in your diet is 0. You need no carbohydrates in your diet. You can eat absolutely 0 carbohydrates. You can make all the carbohydrates you need yourself. Okay. If you just get enough calories. So like grains, the amount of grains you need, the bottom of the food pyramid, the thing they say you should eat the most of, you need none. You can actually go complete without grain. No problems at all. Meat, which is a kind of highish thing on that table, you actually need a lot more of, or something that replaces that meat effectively, right? And then vegetables and fruits. Vegetables is like the primary thing you should get, and it's just a fairly small fragment. And fruits are very high priority as well. Not quite as high as vegetables usually, but yeah. Like the food pyramid is almost exactly opposite of what you want. And the funniest thing about the food pyramid is when they proposed the food pyramid, 14 of the best nutritionists of the day came out and said you're going to create a generation of fat asses and you're going to cause a bunch of health epidemics. Guess what we did? We did exactly that, didn't we? Yeah. They were blacklisted, by the way. It's actually a fascinating story. There's a documentary on it. You guys should watch that sometime, about the people that came out against the food pyramid during the 70s. Yeah, they had their careers destroyed. And they were absolutely right. Provably, at the time, they were right. Yeah. So it's wild. Nowadays, actually, the thing that we had went to before Maha, I don't know if it's still a thing, 'cause I bet you they've taken it down now, was actually a website. So a website that had a health score for different foods that you look up. This was actually implemented on the last presidential term. And that website's kind of funny because it ranks eggs as a 30 of 100 in terms of healthiness. And it ranks Lucky Charms as being a 70 out of 100 for healthiness. So it ranks Lucky Charms as being healthier for you than eggs. It wasn't exactly a step forward. um, some proteins, especially those from plant sources, are what are referred to as incomplete proteins. It doesn't matter how much of them you eat, you will never get all the proteins you need in your diet, and so you always have problems. In fact, you can actually end up starving to death and yet being huge and getting way more calories than you could possibly ever need, right? Because you can eat incomplete proteins until you run out of enzyme that you need and die. Okay? So it's actually kind of interesting that there's a fairly large chunk of people in the United States that are labeled as being as starving, are labeled as being not able to get the nutrition they need, despite the fact that they get way more calories than they need. And that's because they eat a lot of incomplete proteins. That's one of the big reasons. The other reasons is they don't eat things like vegetables or fruits. You know what the only group that still gets scurvy on significant numbers is? Scurvy is in like pirates. Arg. I mean the scurvy. College students. Because college students typically don't eat fruits, right? So you need to eat like citrus usually, vitamin C, and you need to get that every couple days actually, because citrus is water soluble and you urinate it out. Okay? If you don't have enough citrus in your diet, you tend to catch scurvy easily. And you get bleeding gums and all sorts of mm-hmm. And why like sailors are called limeys? Because they used to wear limes on their neck to prevent scurvy, though ironically not so they could eat it. I mean, they had like kind of the cause and effect, but wasn't quite right. Okay, so a complete protein meal can be obtained by combining certain types of foods. Usually, they recommend something along the line of like soybean, eggs, fish, some meat, poultry or red meat or pork or whatever. Okay, depending on your calorie intake you're trying to reach. You can do the whites of eggs to cut down on calories. It won't give you every single protein then, but it still gives you a decent mix. Right? So this is the classification of amino acids. Amino acids are going to be separated in two broad classifications again. That's going to be based on their side chains. That's nonpolar and polar. There's a few exceptions for the side chains of nonpolar amino acids. These are going to be composed entirely of carbon and hydrogen, and they'll typically be hydrophobic. Now keep in mind, there are a couple of exceptions. Not every single one is entirely just a hydrocarbon order. Okay? Tryptophan is going to contain one nitrogen atom. This does contribute some polarity to it, but it's so much more nonpolar in its structure that overall it ends up being nonpolar. Methionine does contain a sulfur group. This is going to be the methio portion of it, and so it has a CH3 at the end, and so it ends up being essentially nonpolar. Okay, though you can make hydrogen bonds to the sulfur, but it's not very effective because again it has that methyl on the other side of it, right? All right. And so this is again your classification of amino acids. Your polar amino acids are going to be further divided into three groups. We have the neutral, acidic, and basic. The polar amino acid side chains are going to contain functional groups that are going to create an uneven distribution of electrons on that side chain. And that's what makes them polar, right? That they have poles, a negative, and a positive side. Okay? And so all the polar amino acids, except cysteine, which the SH, are going to be hydrogen bonding with water because they're going to have OH or NH, right? And so they're all going to be able to make, and they're going to be accept and donate hydrogen bonds. They'll all be able to make hydrogen bonds. And that's what makes them polar. That also makes them hydrophilic. So the polar, acidic, and basic amino acids are going to have charged side chains. This actually makes them able to make ion-dipole interactions and ion-ion interactions, by the way. So you can actually get salting on those portions. And in fact, one of the things that helps out protein structures are things called salt bridges, where they actually make little bridges that look like salts. So kind of cool. And that's because they are charged side chains. So let's talk a bit about the abbreviations. So the amino acids are going to represent the vocabulary of proteins. They're going to be your sort of like letter code for the proteins, right? Your alphabet. And so the amino acids have 3-letter and 1-letter abbreviations. These abbreviations are basically the best thing they could have done for the full name. The full names were created first. Then they realized, oh my God, they're 10,000 long. We need to do something. And so then they created the 3-letter abbreviation and 1-letter abbreviation from those full names. Now, because those full names were created first, they ran into a problem. There's a lot of repeats, aren't there? If you look at the names, they have very similar names, don't they? And so you can only use A one time, right? You can't use it over and over and over, can you? And so what we end up getting is the 1-letter abbreviations sometimes have nothing to do with the name, okay? And that's because we just couldn't use the 1-letter abbreviation again, right? We ran out. So of the 20, 11 of the 1-letter abbreviations are actually going to start with the first letter of your name. Ideally, they would have all been that way, but we couldn't have done it, right? There's repeats for the full names. The remaining 9 amino acids have 1-letter abbreviations that start with different letters because we had to do different letters. Table 10.1 actually gives you the origins of these abbreviations in case you would ever like to know. This right here is your amino acid. This is your 3-letter code for them. The 3-letter codes, for the most part, are pretty good. Like very, very, very similar to the names, right? The 1-letter code are these right here. And this one is going to be one that starts with the letter C, H, I, M, S, V. Those are always the ones that start with the letters. This is going to be the most common amino acid that starts with that letter. And so this is the A, G, L, P, T. These all start with that letter. And that's because these happen more in proteins. And so the one you see more of, they gave the letter. Which makes a lot of sense, actually. Arginine, on the other hand, ARG, is going to be R because that arginine, has a very hard R, doesn't it? And so it's kind of easier to remember. The easiest way to remember it is actually R, like a pirate, right? ARG. Me mateys. This one's phenyl, fen. That's the sound of F, isn't it? And so they gave it F. Y, tyrosine has a Y in it, and so they gave it the Y. Okay. Tryptophan, you'll notice if you say that fairly quickly, or in certain accents, it actually has a very W-sounding name, doesn't it? Twip. Can you hear it? Yeah, so tryptophan, twip. W. This one right here has a hard D sound in it, and so aspartate, D, and they give it a D. Arginine, you can hear the N, can't you? Very hard N. Glutamate, the glue here, you actually end up in English having to insert an E into that sound. And so that was given an E. Glutamine sounds a lot like Q-tamine. And so they gave that one a Q. Honestly, this is where they end up kind of... I get what they're going with. But, yeah. Doesn't actually help, does it? This one right here is lysine. They literally just went, I give up, and it's the next letter down on the alphabet. There really isn't anything good there, right? What are you going to give it here? I gave Y. So. Let's look at the weak acids here. So all amino acids have an amine and carboxylic acid functional groups. They're going to have charged and uncharged forms. So if you get them at the correct pH, they're going to actually be on charge. If you get them at physiological pH, they tend to all be charged with charges on positive charge on the amino, negative charge on the carboxylate portion. The amino acids are going to exist in different acid-base forms depending on the pH of the solution. They're all going to actually create their own buffer systems if you have enough of this concentration. And so they all are going to generate a little bit of a buffer system. The amino acid alanine actually is shown in its form that predominates at the physiological pH, which is 7.4. The ionic form that containing no net charge is called the zwitterion. This has no net charge, but it's going to have a positive and negative end on each side. And so that is the zwitterion, literally meaning Zweihander, two-hander, Zwitter-ion, two-sided ion. Okay. Each molecule has a unique pH value at which only the zwitterion is present. This is actually called the isoelectric point, and it's denoted as P sub I. You'll recognize the P from like the pH, PO8, and then I for the isoelectric. Okay. The Pi of alanine, the negative charge on the carboxylate ion is going to be balanced by the positive charge in the ammonium ion. Your net charge is 0, and that's this right here. So if you were to add acid to this, what you would actually see is you would actually start putting charges on the carboxylate portion of that. See that? And you'd actually disrupt this as being a zwitterion. Only have one charge, positive. Okay. If you were to add base, on the other hand, you'll deprot the amino portion, and again, you'll get a negative charge one. Okay. And so you can actually disrupt that isoelectric point. pH 6 is actually where the isoelectric point is for this, where you'll have equal amounts of both form. This right here is where you would have the majority of this form. So the charge of your functional group is going to change as your pH changes. If you sat there and just put acid in this, you'd actually see the charge and which form you have more of change as you added acid. Okay. At pH values below the P sub I, the isoelectric point, your acidic form is going to predominate, and your functional groups, which are the protonated ones, are going to be the predominant form. And so what you'll see here is at pH 6, you would have the majority of this in this form. Okay. As you add more acid, what's going to happen if it goes below pH 6 is you're going to get more of this form. Okay. And so you end up with more of this form. If you are higher than pH 6, like 7.4, you're actually going to end up more with this form. And so what you see at pH 7.4 is you're still slightly more of this than this, but there's a lot of this in there. Okay? Okay. So we actually studied condensation reactions back in chapter 5. You remember that? Where we removed water from it and we linked them together. And so this is actually another type of condensation reaction that happens whenever we form protein. Okay? This is going to give you a dipeptide once you put two of them together. Peptides are actually another way of saying the joined monomers in a amino acid after amino acid after amino acid. Okay? It's another way of saying the amide bond is a peptide linkage. It just is another terminology. So the carboxylate ion and the amino acid, the amino portion of it, are going to join on two different amino acids, and they're going to form a dehydration or condensation reaction where you remove water molecule, and this is going to give you an amide functional group. Let's go ahead and look at the reaction real quick. So this right here was that condensation reaction we did back in chapter 5. We had carboxylic acid, we had an alcohol, they reacted together, and they gave us water, right? And the ester in that case. If we look at our amino acids, right, here's an amino acid reacting with another amino acid. to create a dipeptide or a mid-linkage. And so this is the carboxylate on one amino acid reacts with the protonated amine on the other. This is going to be a condensation reaction, so water's going to come off, and we'll be left with the amide right here. Notice that this part right here is the amide functional group, the one in green. Okay. This right here, by the way, was the sugar ones that we were looking at before. This was also a dehydration reaction. So this was the hemiacetal. That was the ring-closed form of the sugar. And this is what we would have been looking at in terms of making the acetyl, where we would join it to make like glucoside, or we would join them to make like starch or any of those things. Okay. And so that was also a condensation reaction, like we had looked at. Okay, so this is the amides and the dipeptides forming the dipeptides. And so this is, again, a peptide bond when you make it. So we do our condensation reaction. What we see here is we have the carboxylate reacting with the amino on two different amino acids. In this case, we actually labeled them. So this is alanine and valine. See, there's the side chains. That's what makes those amino acids. Okay. Notice this part of the structure is the same for all, right? Okay, so we do a condensation reaction here. What we notice is that we actually have directionality to this amino acid, right? So this dipeptide now has directionality. It has one end where it still has the amino group, and the other end is going to have the carboxylate group. Okay. These are referred to as N-terminus, that's where the amino group is, and C-terminus, that's where the carboxyl group is. Right? And so whenever you're looking at proteins, they always have two different ends. They have the N-terminus and the C-terminus. And that's all that means. The N-terminus is the side you have the amino exposed. Okay? The C-terminus is the side you have the carboxylic acid exposed, and typically where you join more of them to it if you're going to join. Okay, so in a dipeptide, your N-terminus, the amino terminus, this is going to have your unreacted alpha amino group. The C-terminus, the carboxyl terminus, is going to have your unreacted carboxylate group. By convention, you write the N-terminus on the left side and the C-terminus on the right side. Here's the identifying the components of your peptide. So when you're looking at the components of your peptide, an easy way to sort of go through and systematically identify what you have is to circle the amino acid at the N-terminus, give its name. The next thing you would do is put a square around the amino acid at the C-terminus, give it its name, and then you'd locate the peptide bond by drawing an arrow to it, and that would give you your peptide bond. You give the 3 and 1 letter abbreviations for this dipeptide. 3-letter abbreviations is more common, because at this point you don't really need to use the 1-letter abbreviations. But here's an example of looking at what this would look like. So again, dipeptides are going to be formed with 2 peptides, 2 monomers, and so that's going to be 2 amino acids. Each pair can form 2 different dipeptides depending on which is at the N-terminus, which is at the C-terminus, right? Which one's first, which one's last. The 2 dipeptides are going to have structural isomers, that is, different compounds that give different properties. The order of the amino acids is actually critical to its structure and the function of the compound. It's not the only thing that's important, but it is the first step that you have to understand a protein at all. Okay? And so, right here we have the N-terminus for both these. This one has alanine at your N-terminus, and then valine at the C-terminus, right? That's one we saw before. You could have had the other isomer of this that won't give you the same dipeptide, that would not give you the same protein if you were to do this, give you something different, okay? And that would be where your N-terminus has valine, your C-terminus has alanine, right? And that's because they're flipped. In this case, because you have an N over here, an amino, and our carboxylate over here, these are never the same thing. They're not symmetric, right? They have a direction. That making sense? Kind of like cars, right? Not the same thing to back up along the street. So any compound contains amino acid joined by a peptide bond can be called a peptide, of course. Dipeptides are going to be your smallest peptides. Compounds with 3 amino acids are called tripeptides. 4 would be tetrapeptides, and you can actually go on and on with this. As the number of amino acids increases, you typically give up on naming these off. You can actually name these up into the hundreds, actually. This naming methodology just keeps going and going and going. So you could do it for infinity. But typically you give up somewhere around like the 4th or 5th. Okay? And you just call it polypeptide, meaning many, right? Biologically active polypeptides contain 50 or more amino acids as a protein. And those are where you sort of start moving from polypeptide into protein. Is because at 50 or more, you're typically a full protein at that point. The polypeptides are usually when you're talking about fragments of proteins that have not yet been fully assembled, or maybe you broke a protein apart and you're looking at the fragments or something like that. So let's talk about some problems. So peptides can cause problems in celiac disease. The substance gluten, excuse me. Okay, so gluten, this can be a mixture of proteins found in various grains. They can include wheat, primarily used as a structural molecule so they can grow taller. Okay. The 2 main proteins of gluten, glutenine and gliadine, are going to contain rich regions in the amino acids proline and glutamine, which you may notice as part of these namings right here. This is going to be a unique amino acid sequence. When gluten enters the digestive system, some of the peptide bonds are actually going to be hydrolyzed by enzymes. These are called proteases. Their primary purpose is to actually break up proteins, so you can harvest them for their amino acids. Obviously, you don't necessarily want to take in the full proteins of another animal, because then they would do other things, right? Or even plants for that matter. These again are going to break down gluten. They're going to leave behind the proline and glutamine-rich peptides in your digestive system. Some of the glutamines in these fragments are going to be modified by glutamate. This will form an enzyme that's classified as a glutaminase. Notice the glutamine portion right here, ACE meaning an enzyme that acts on it. This is going to change the side chain in glutamine from an amide, right there, to a carboxylate group, which is that COO minus. And so that's what it would look like. So right here, glutamine, glutamate. And this is the enzyme that does that change. So that transition here is going to change a polar neutral residue to a polar acidic residue right there. The addition of the negative charge to the peptide sequences actually causes the people with the autoimmune disorder, celiac disease, to launch an immune response against these peptides, and so their immune system starts attacking those. Does anybody know what all the side effects of... does anybody ever had the flu? Or COVID for that matter? Okay. Now, what parts are the flu doing it? Is the fever because of the flu? No. What is it from? Your body controls. That's your immune system, isn't it? Yeah, your immune system's trying to kill the virus, and so it's doing that, isn't it? Even the coughing is actually your immune system doing it. So very few of the things that we associate with diseases are actually the disease doing. Most of those things that we associate with them are actually our immune system trying to fight it, right? And so in autoimmune diseases where you're attacking something yourself, or in diseases where you're attacking a fragment of something that's not even alive, that could cause a lot of damage to you, right? Because they're going to be fighting that. They're going to be doing all these side effects, and you're going to have just rampant side effects the entire time, right? And so these can actually be incredibly damaging to you. In this case, you're going to be attacking these fragments right here. So some of the symptoms of this are going to be inflammation of your small intestine. That's that puffiness you get whenever you have an immune response. That's inflammation. There's a shocking number of diseases that are linked to inflammation now. That's been like the key word in the medical community lately. Inflammation. The lower intestinal absorption of nutrients, so you get very low absorption in your intestine for the nutrients. This can cause malnutrition, and so you're going to have not the things you should have been absorbed. You're going to not have those in your system, meaning you won't be able to produce the sort of enzymes and proteins that you should be producing. People with gluten sensitivity should avoid gluten in their diets, of course. They can have a very small amount, and it'll cause very small amounts of inflammation then. But if they get too much, obviously they're going to have major inflammation. They're going to have problems that will put them on the bathroom stool. Okay. And they also will have problems getting the nutrients in their diet they should. And so long term, it can be extremely problematic. Anybody have a gluten allergy? No? Oh, that's actually kind of unusual. It's not an uncommon allergy to have. So let's look at the primary structures of a protein here. So when we look at proteins, there's different levels we need to understand them on. The primary structure of a protein is going to be the order that the amino acids are in. This is an easy way to think about this, is it's the protein backbone. Okay, it's the order of the amino acids in that protein backbone. You would typically list it from the N terminus going to the C terminus. So the N terminus comes first on the left side in English, and then we go through to the C terminus. We do it as a straight chain because we don't really care about anything else when we're talking about primary structure. The side chains of your amino acids are going to be substituents that dangle from the backbone when you're looking at the primary structure. By convention, you typically try to list them on top, but if you run out of room, you can flip them to below. Okay? And so that's the primary structure of a protein. Notice that does not give you the entire information about the protein. That would be like looking at a skin of a car, right? Doesn't give you all the details about a car, does it? Okay, let's look at the secondary structure now. So the secondary structure of a protein, this can be involving regions that can form coils or other repeated patterns of structure within your three-dimensional shape of the protein. So we're kind of zooming in and looking at the little patterns that develop over and over. If you were to compare this to human anatomy, that would be looking at like the organs, right? If you were to compare it looking at a car, it'd be looking at the different parts of a car, right? So the two most common secondary structures for proteins are going to be the alpha helix. That can be named, both of those are the exact same name, by the way. That's just the symbol alpha here. And the beta pleated sheets. And that's the symbol beta right there. So beta pleated sheets again. These are the two most common structures that you would see in a protein. They're not the only secondary structures, but you got enough of it if you know those. You can kind of get by. The secondary structure in your protein are going to be stabilized by hydrogen bonding along the protein backbone. And they're going to involve amino acids that are near each other in that primary structure. They can also involve hydrophobic interactions between those side chains. Okay. So if we look at chapter 7 again, we have the hydrogen bonding. This is not a covalent bond, but it's a very strong attractive force. And so what you'll see here is either large amounts of nonpolar attractions or hydrogen bonding can hold things together quite effectively, actually. Okay. Is it completely uninterruptible and we can't do anything to it? No, that's not true, right? And we know that because what happens if you cook an egg? It cooks, doesn't it? Yeah, it changes. And what you're doing is you're actually breaking apart the structures in the proteins. Okay? That's assuming you don't burn it, in which case you just break apart the entire protein. All right. This is an example of an alpha helix, a secondary structure right here. And so we're actually seeing the alpha helix here. This is a cartoon representation, which makes it a bit easier to see. And notice what you see is hydrogen bonds coming down repeatedly over and over, and that's what holds it in that helix structure. Okay. Now, what I would like to point out is a very common misconception for these alpha helix structures is like they're tubes, they're straws or something. They're not. This has no empty space. Okay? If you were to think about these atoms right here, they're filling up that entire space. All right? So they are not actually tubes. This has no open space in it. Okay? It's just a helix that comes up like a rebar helix. Okay? And that's actually about what it's used for. So largely a structural component that makes structures that you can do stuff with. Okay? So again, the hydrogen bonds are going to be positioned in such a way that they connect the repeat units one on top of the other in this helical structure. This right here is your beta pleated sheets. There's actually a few varieties of these, but we don't need to go super into detail on that. Right here we can see where they're moving the opposite direction here. See that? And so they wrap back around like so, and they line up perfectly with the hydrogen bonds being nice and straight. Okay? And this is one type of beta pleated sheet. And it's sort of the easiest to understand and look at. Okay? And they literally end up making something that looks like a sheet. And it has, everybody knows what pleats are, right? So everybody's done like an accordion. Everybody's seen an accordion or paper. They fold it back and forth over and over. Okay. That's what this looks like. So it comes down, up, down, up. Okay. And these are little turns. These are still part of the protein right here. They're just turns in the protein. So this would be like usually four or more amino acids that make this turn. Okay. All right. So let's talk a bit about protein secondary structures in Alzheimer's disease. So Alzheimer's disease, very common form of dementia. This causes memory loss and the ability to think clearly, becoming severe enough to interfere with daily tasks. And actually, you end up having parts of your brain die during this process. So it is eventually lethal. Though usually people don't actually die of the Alzheimer's disease itself. Okay? Because typically they're elderly as well. The disease is progressive, most often occurs in adults over the age of 65, though you can have early onset dementia and Alzheimer's. Okay, that is a possibility. People with the disease have distinctly different brain tissue than people without the disease, though it's actually related to a number of other conditions that are also very similar to Alzheimer's disease, okay, where they develop plaques. Your brain tissue of people with Alzheimer's disease actually contains plaques between the brain nerve cells. The buildup of... Now, this is actually something that they just recently have had a bunch of research coming out. And this has been partially debunked. Okay? So I'm going to give you the standard explanation for this. But Alzheimer's disease looks like it might be actually really complicated exactly what caused it. People are less sure now than they used to be. Might actually be several causes. The buildup of plaques between neurons and neurofibrillary tangles inside the neurons are thought to interfere with nerve impulse transmission and contribute to neuronal cell death. The more recent research actually indicates that this might be a very similar process to like a diabetes of the brain. And it's actually a lack of control of the sugars that might be involved in this. But there's been a bunch of new research coming out, and some of them with actually very promising results for being able to at least delay or possibly even treat Alzheimer's. So, let's talk a bit about the protein structures, which is definitely a component of Alzheimer's. So the protein plaques are going to be made up of protein fragments. These are going to be 42 amino acids long. These are actually called beta amyloid protein. You may have heard of that if you've ever heard of Alzheimer's, the beta amyloid proteins. The protein sequence is going to be identified in both healthy and diseased individuals. And so this is a naturally occurring in your body, but typically it doesn't build up plaques in your brain, okay? Unless you have Alzheimer's. In a person with Alzheimer's, the structure of the beta amyloid is going to be an alpha helical. But in a person with Alzheimer's, the beta amyloid proteins actually form beta strands instead of the alpha amyloid. So usually they make helical structures, and that keeps them from forming plaques because they can't nicely associate with each other. But when they form sheets instead, they can then nicely associate and they build up, and they can form plaques then. Again, that's going to make them fold into sticky beta-pleated sheets. These will then aggregate. They're going to form plaques at the neurons. Did anybody notice that we're using several different layers of chemistry knowledge that you built up to this time? Non-polar associations happen better when you have larger surface areas, right? Oh my goodness. It's like you have some knowledge now to be able to do stuff with it. Getting your money out of this course, aren't you? Okay. And so right now there is no cure for Alzheimer's disease. There are medications that can have some degree of effectiveness on it. They can slow its progression, lessen symptoms, and they can counteract some of the side effects of it by otherwise making the brain's transmitters run at a higher function. But there currently has not been actually any cure for Alzheimer's disease, though interestingly enough, there's dietary regimens you can go on that actually can halt the progression of Alzheimer's for limited periods of time. So there's actually been some interesting work recently coming out over the last 2 or 3 years. These are right here are the alpha helical and beta sheets. Everybody can see the plaques sticking together pretty nicely, right? And why it happens. Yeah, these can't stack up nicely, can they? No. So they have very limited association they can do with each other. These, on the other hand, when you have beta-pleated sheets, they can associate very nicely, can't they? And so then they stick together, and they form the plaques. This is the beta amyloid plaque right there. Okay. Let's talk a bit more about the tertiary structures now. So the alpha helices and beta-pleated sheets of the polypeptide chain, they're going to interact with each other in the environment to create what's called the tertiary structure. And so the tertiary structure is that three-dimensional structure of a single polypeptide. Okay? Now, some proteins are assembled of multiple of these. And so that's going to be the next step up, is where we have multiple chains that associate together. But if you only have one chain, all you have is the tertiary structure right here. Okay? And that's as far up as it gets. And so things are going to fold back. They're going to make these different structures, and that's your tertiary structure, your overall three-dimensional structure of a single polypeptide chain. Your nonpolar side chains are going to be repelled by aqueous environments, and they're going to turn towards the interior of your protein, of course. The polar side chains are going to be attracted to aqueous surroundings. They'll appear on the surface of your protein and allow it to interact with the water around it, right? Now, note that if you are in a nonpolar environment, such as the cell membrane, this is going to be inverted, right? So the nonpolar parts will interact with the nonpolar cell membrane, right? Tertiary structures can be stabilized by attractive forces between side chains and the environment, as well as attractive forces between side chains themselves. All of these computing interactions actually create the three-dimensional shape that your protein's going to stay in. Though, interestingly enough, proteins don't necessarily fold back into their original configurations if you unfold them. A lot of that has to do with how it comes out as you make it, and different things in the cell that can help them fold. Okay? And so the actual structure that you should have for your protein is held in place by these forces, but doesn't necessarily fold itself all the time. Okay? Some of them do. Some of them are small enough and can fold themselves. This is what you would be looking for as a tertiary structure. And we can see the overall structure here being made, right? And all the different forces happening. There's those bridges I was talking about. They have an ion-ion interaction. Hydrogen bonding right there. Hydrogen bonding, these are hydrophobic interactions. These are those disulfides like you'd get for curly hair. Okay? Which is thiol-thiol interactions. Okay? Next time, we're going to pick up right here where we'll go ahead and define the different interactions in tertiary systems and talk a bit about quaternary systems. We're actually well on time, aren't we? You all have a wonderful day. I will see you around. Anybody know when the next exam is? Finals week. Yep. You got it. Don't forget to check out the next exam. We won't have any lecture times that week. So don't forget to check out the actual time for the exam. Oh, okay. Could you send me an email to remind me? I'm not going to be able to do anything about it until 7 to 9. Yeah. But I'll modify the date. Okay?
I scheduled the Wednesday.
Okay. Sounds good.
Thank
I'll
you.
see you Wednesday 1:30.
Perfect. Thank you.
Oh, I'll only be able to give you an hour though. Okay. Yes, that Friday before?
Yes.
You got it. Yeah. Yeah. Come on at 9 a.m.
9 a.m.
Mhm.
Thank you so much.
I'll have you take it along everybody else.
Thank you so much.
Yep.
I'll see you then.
I'll see you in Lab and D. We'll have some fun with acid space. Yeah. 39.